Good morning, good afternoon, good evening. Thank you very much for joining our MVP demo event. My name is Stefan Harrer. I'm the director of the AI for Science program at Australia's National Science Agency, CSIRO. Some of you are joining us from around the world in other time zones and so won't be able to be live in here. For all those of you who are watching the recording, the slide that you see up here is your run sheet of what's in the meeting. And if you decide to fast forward to certain parts of the meeting, there's your timestamps on there. I would like to begin by acknowledging the traditional owners of the lands we are meeting on today and pay my respect to their elders, past and present. Exciting things have been happening at the intersection of science and AI lately. Just a few weeks ago, we had Lila Sciences come out of stealth mode, raising 200 million in a pursuit of scientific superintelligence program that sets out to empower researchers in the health and life sciences disciplines with autonomous labs and AI-powered assistance for their scientific discovery work. Google has come out with an AI co-scientist, also very recently, that aims to accelerate drug development and again assist biologists and researchers and scientists in the health and life sciences with everything they do in their hypothesis generation and ideation work. And last but not least, I had the pleasure to listen to Kimberly Powell, NVIDIA's VP of Healthcare, and what she shared at the NVIDIA GTC just a few weeks ago was mind-boggling. NVIDIA unveiled a set of so-called AI agents, assistants for labs, clinics, and trials that focus on turbocharging what scientists and health and life sciences researchers can do using AI as part of their daily work. This is a trend, and a trend we here at CSIRO are proud to have actually helped to set. The idea is that we can use AI and put it in the hands of scientists to turbocharge and scale the scientific method. We all know that the scientific method is one of the, if not the most important accomplishment of humanity. We have an idea, we set a hypothesis, we then design experiments to test this hypothesis. We run these experiments, we look at the outputs, we analyze that data, and we then make a call on whether the hypothesis held up or not. And depending on what we find, we do this over and over again. That scientific method will not change. That is the blueprint of how science is done. But what we can do is we can infuse AI at every single step in that process and turbocharge and scale it. This is essentially applicable to every scientific domain. But for reasons we'll get to in a second, the health and life sciences, biology, genomics, have been primary landing pads for bringing this new AI-powered capability to scientists. The lay of the land looks like this. We have a few expert bioinformaticians, AI scientists, AI engineers who understand, have possibly even built, and can use and access advanced AI tools to do part of their research and scientific work. The bulk of biologists, the bulk of researchers in the health and life sciences does not belong into that cohort. If you look at the numbers, we only have about 18% of biologists who currently use AI as part of their work. Over 80% of biologists out there do not do that. They are cut out of the immense potential that comes to scientists when they have the ability to infuse AI into their scientific work. We want to change this. We want to make AI a commodity. We want to empower all biologists to use AI without having to be AI experts. So this is what we here at CSIRO have worked on for the better part of the last year. Our goal and mission is to harness the power of not only, but very predominantly generative AI to create a scalable, a secure platform as a service that brings AI in form of assistants, so-called agents, to labs and researchers. We want this platform to be both a landing pad for contributions of scientists who have built models, who have created data sets, who have created agents and infused these into the platform, make it available to the world of scientists out there. And we also want this to be a landing pad for users, for scientists who want to access these AI models and tools and data and embed it in their scientific discovery workforce. This spans the wet and the dry lab. It's very important to validate what scientists come up with in terms of hypothesis modeling in the real world. So these assistants that we are building and that we are testing and using cover both the physical and the digital world. Ultimately, we believe that by making these AI agents available to scientists, we can spark and increase their productivity and their creativity. We can help them to speed up the path to scientific discoveries, which would be otherwise have been decades away, out of reach entirely. From being more productive and being faster to being more creative and having new ideas, we think AI can help that process dramatically. And that's why we have built, and we'll show you in a second, the Agentic AI platform for scientists. Think of what we're doing here as giving scientists the glue that brings it all together: data, tools, models, and the interface to users that's seamless, that's easy to navigate, that allows this ecosystem to be used and governed by AI experts, but as well by non-AI experts. The reason why this has been so exciting and why we chose biology in the health and life sciences as a first area of co-design, demonstration, and testing lies in the fact that all these elements that I just referred to, data, models, and tools, have undergone a profound revolution and transformation and, frankly, explosion over the last couple of years in biology. I just want to highlight a few significant points, voices in the field. Here's Eric Topol's paper on highlighting the diversity, the size, the messiness of data that scientists in biology, in the health and life sciences have to deal with, seek to analyze and make sense of, to decode biology. The same is true for the advancement of tools that become rapidly available to scientists, either because they built them themselves or because others in the field built them. That goes for both the wet lab and the dry lab. It would be too much to go through all these tools and features that I've listed here. As a matter of fact, I'm borrowing here from, again, Kimberly Powell and her GTC NVIDIA talk. It illustrates very, very profoundly that we're looking at a hockey stick curve here of an explosion of variety and capability in both dry lab and wet lab models for biologists. Very important, again, we need to span this bridge between wet lab and dry lab in what AI assistants, AI models, AI agents can help with. And this is our mission. This is what we have worked on. This is what we are still very much working on. We've come to a point where we have built an MVP that we'd like to share and demo to you. But without further to say, I will now hand over to our head of product, Regina Campbell, to tell you a little more about this MVP, and you'll see more demos and information on it throughout this whole presentation. Over to you, Regina.
Thank you, Stefan. As Stefan said, I'm Regina Campbell, and I am the lead product manager on our science digital platform. and I really wanted to give you a quick introduction to the beta platform demo that you'll see in just a few minutes. Our beta platform is built to showcase the foundations of scientists and researchers working collaboratively with generative AI on meaningful scientific challenges. Today, you will see two demos. One, an overview of the platform capabilities, and the second is a live demo that will show you how biologists and genomicists can work to identify enzymes with specific functions. Whilst the platform has these domain-specific capabilities, it also has a variety of domain-agnostic features, such as deep research or hypothesis generation assistance. Before we dive into those demos, I also want to call out some features of our platform that you may not notice in the demo, but are critically important. Our guardrails and responsible AI are built into the experience and continually improved upon. Through our beta testing, we are also exploring how to best present the accuracy of these results and the thinking that the LLMs are doing. We are cloud provider agnostic, meaning that we can adapt to any cloud provider and utilize multiple cloud platforms together. We're adaptable to a variety of agents. We also have a meta agent built in, which acts as your primary contact point, beginning with offering recommendations on tools and agents to use. And this is expanding into what we call asking, where you can tell the meta agent what your goal or objective is, and it can present back to you the tools and approaches you might take. It will also be able to handle increasingly more complex questions. Our platform is also aiming to bridge that physical and digital worlds. Right now, that means working towards a connection to the biofoundry, but in releases later this year, it means integrating with robotics capabilities and expanding into other lab environments. It's critical to say that today's experience is focused on genomics, but the platform is not a genomics-only platform. Engineering product design teams working closely with scientific research teams who have or are building capabilities that, when utilizing the infrastructure agents we have developed, enable these experiences. I'm excited now to share these demos with you all.
Let's use AI to supercharge science. Introducing CSIRO's latest digital innovation, an agentic AI-first operating system that's transforming how scientists work by seamlessly connecting physical, digital, and social systems, accelerating discoveries that once seemed decades away or entirely out of reach. Built by scientists, engineers, and AI product designers, this system helps you create and execute intelligent workflows that respond to science domain agnostic and to science domain specific requirements. Tell the system what you would like it to assist you with, and the AI agents will handle the legwork. From hypothesis generation to experimental design, from the virtual lab to the physical lab, brainstorm, verify, and refine new research ideas. Design, run, and simulate experiments and analyze data to gain new insights and build knowledge. Working with AI agents will empower scientists to make discoveries at an extraordinary pace without having to compromise the scientific method. The system can even spark entirely new research ideas, opening doors to discoveries that might never have even been explored. Our MVP is built for and by researchers in the health and life sciences, supporting digital and wet lab work. This is just the beginning. The system is already assisting scientists across disciplines as CSIRO researchers test, expand, and harness its capabilities to accelerate scientific discovery. Designed as a fully integrated platform, the system is purpose-built to support scientists, embedding key scientific best practices like data privacy, transparency, reproducibility, explainability, reasoning, and human oversight. It's responsible by design, with ethical and safety principles embedded at every stage, from data handling to decision-making, and a strict adherence to the scientific method. CSIRO is your trusted partner in innovation. We welcome opportunities to showcase and grow the platform's impact. Reach out if you'd like to learn more or explore a partnership. Please email sciencedigitalprogram@csiro.au.
Thank you so much for that, Regina. My name's Rahul, and I'm a genomicist, and I work on primarily product development, as well as developing the use cases for the platform. I'll walk you through a live demo of what you actually just saw. Now, just to sort of take a step back and actually think about what we are looking at. The first paradigm shift that we saw with large language models was the innovation of chatbots, things like ChatGPT. This allowed people to access information a lot easily. The second paradigm shift that we saw from 2024 onwards was actually the innovation, was like the onslaught of agents, really, the era of agents. And think of each agent as essentially driving its own chat GPT, and the agents can actually talk to each other. And that actually allows us to create a much more significantly complex and steerable mechanism and steerable system. And this is actually where a large language model start becoming a lot more useful for researchers who are not actually developing the large language models themselves. And the second paradigm is specifically what we're aiming to make easily accessible to scientists so that they can develop and use agentic workflows. In a manner of thinking, you can almost think of this as we've turned these agents into Lego blocks. And what we want to do is we've built a platform where users can actually come and build those Lego blocks, whether they are agents, whether they are analyses, whether there are any specific tasks that they need to do as biological research, biologists or non-biologists or what have you, and actually get complete entire projects within the ecosystem. And it is entirely up to the user or the scientist's creativity to actually turn these Lego blocks into something quite beautiful. And we've started off in the MVP with building a few very specific kinds of blocks. They're either kind of query tasks, such as finding experts or overviews or doing deep research into specific areas, patent searches or what have you, or analytics, such as finding novel genes, finding the functions of certain genes by doing an extremely deep and comprehensive analysis. Or finding the structure of proteins and then and using those in specific analyses. Now, if you think about a standard scientific project, that usually can be broken into a couple of different tasks. These tasks can either involve, are usually around the ideation phase, the review and finding collaborators and so on and so forth. And that generally then distills down into actually writing grant proposals, designing experiments, and then doing the actual science. And so that usually splits sort of, this is sort of how it looks. And the critical component around that we've built the platform around is creating that interdependency between tasks so that information from one task can flow into another. It's a standard drawback with basic ChatGPT-like interfaces, which act fast, they act quick, but it is usually hard to get them to change directions entirely. But with the way the system has, the way the platform has been built at the moment, it allows us to run discrete tasks and actually get one task to learn from another task or a few other tasks to actually do a job well. And each of these are essentially complex agentic systems. For the purposes of the demo, we'll actually go through a much simpler and leaner version of this, where we'll actually pick a research question. We will get a topic overview on it, identify gaps, do a bit more research into it, and actually go down some analytical workflow that Gunjan will then take you through. The question that we actually want to address today is looking at lignin degradation using bacteria. Now, lignin is a complex organic polymer. It is found in cell walls. It is also one of the sort of most significant byproducts of paper manufacturing, if you will, and hence is a very significant pollutant or a biological waste, which we have been looking at using as a carbon source for the longest time. And if we manage to do that, that's an extremely important breakthrough in sustainability. Now, studying lignin directly is quite difficult. It's a very complex compound. It has a very complex structure as a polymer. And so we tend to use pinoresinol, which is a lignin that is used as a proxy for lignin degradation. And it has it's much more easily found, and it can actually be used in the lab. And so just looking at the flow that we had earlier, right? In the first instance, when we're designing a research project as a scientist, you want to evaluate the problem, you want to set up the scene. And then you actually want to go through the analysis for the same. So the first one that we will go through, let's say, is the topic overview that we would like to do. Now, before I jump into the platform, I just want to remind everyone that this is actually not ChatGPT, right? What we've done is there's multi-agentic workflows, lots of ChatGPTs talking to each other, in this case, 3 agents that actually do the job. So it takes a lot longer than ChatGPT takes to actually produce the answer, which is why I've actually run a few of these morning this morning so that we're not actually just sitting around waiting and twiddling our thumbs. But it works well with high-level queries, right? So if we jump into the platform, I've already logged in, and we'll resume a project that I had already created. So we're lignin degradation by bacteria, and we want to identify, sort of look into, we can always create new workflows, and you can name the workflows whatever. You want each workflow is compartmentalized from the other workflows so they can be independent. There's a level of independence there as well. And you can start looking, it's a workflow that I just built based on the example that we saw, that we're following today. So we wanted to look at a topic overview, in the first instance, to get a comprehensive overview of the literature on lignin degradation by bacteria. And like I said, one of the key features of the platform is actually being able to give any specific tasks the inputs from earlier tasks. And we've maintained that level of sort of that silo between tasks so that we can have each task being completely different. For example, you can have a market research task feeding into a grant proposal task or what have you. And we'll see an example of that next as well. And so these sort of, they're designed to produce outputs that are more focused towards whatever the need is. And so we have certain subtasks over here. We'll be looking at literature review, that includes finding publications, and then analyzing those results and producing those. So each of these sort of agentic tasks actually give us a comprehensive report, which then the scientists can use. And so from the topic overview, we get a pretty detailed analysis of the gaps and the current state of the knowledge, which is kind of what you want to do when you're entering any new space. And traditionally, so this is just the entire report pasted into a slide, really. And bear in mind, this also depends incredibly on the model that the user eventually chooses, and that's a feature that we will introduce into the platform soon as well. But the next step that we tend to need to do is, once you've got a basic topic overview, you want to start diving deep into the literature and starting to automate that literature review component of it. And so what you would essentially do is use a simple prompt. Like I said, high-level prompts work really, really well with these systems because they're designed to steer according to the input from earlier tasks as well. And so you can just say use an about task as inference, and I actually gave it the earlier task as an input, and get it to actually start extending the overview and paying particular attention to referencing and so on and so forth. And so it does a pretty good job. And Gunjan, being an expert in this area, has promised me that this is actually sensible, which is great. And it produces a pretty detailed sort of report. Now, these can be quite expansive. There have been some such situations where we've got 20-page reports, and in some situations where the answer it can be quite crisp and you'll just get half a page report. But it entirely depends on the topic that you're playing with. And the next step that we tend to usually need to do after that is looking into pattern searches, for example. Now that we have done a topic overview, we have done a bit more sort of literature review on it. We want to know what the actual patent landscape looks like, whether it is something that scientists can look into, or whether you need to collaborate with someone, if there's a competing priority or what have you. Again, we give earlier inputs, earlier tasks as inputs, and it will take those into account and actually then start looking at pulling out key patterns, technology gaps, and so on and so forth. And these can also then, the user can actually then extract and interact with these as well. So all outputs are actually editable, so that we are not restricted to only what the large language models produce, but the users can actually introduce their own insights and knowledge as well, so that any subsequent task then actually takes that human interaction and the human in the loop into account. And just for fun, through the grant proposal thing in earlier, and obviously writing grant proposals is a complex thing, but what helps a lot in many cases is getting a template. And that's the other thing that we can do, is you can give it all the other tasks as inputs, and get it to produce a grant proposal template. And for those with a keen eye, please bear in mind, this is a weaker model that we've used for testing purposes, and there are much high quality large language models that can also be plugged in that significantly improve the quality of the output. And so essentially, if you think about just going back to that space, right, we've set up the project now. We've actually set up, got an overview. We've actually got some background information, and we started designing some of the experiments. And so I got it to, just take a couple of the topic overview on the deep research as an input, and actually got it to start designing experiments, and it will start giving you again, all the, what equipments do you need? What are the materials and reagents? What are the software resources? What are the basic projects that you can, what experiments can you run? And so it really is designed to act as a sidekick for the scientists so that the scientists can actually continuously process lots of orthogonal sources of information before they actually jump in. And I'll actually just pass on to Gunjan because he's going to walk us through how the actual analyses can be done as well, because that's the whole idea behind the platform. It's not just about taking all the AI generated content and actually massaging it into different formats. It's about making sure that we can run analyses and collaborate with the AI driven agents, large language model driven agents, so that we can actually do tangible science. So over to you, Gunjan. Please just ask me to switch slides when you need me to.
Yes, thank you. I'm Gunjan Pandey. I'm a largely wet lab-based scientist. And I'm going to show you 3 bioinformatics tools that we have developed in this platform. And before I talk about the tools, I'll continue from Rahul's story. So through Rahul's approach, he has developed a nice project and got it funded. And he has done some wet experimental work. And let's assume that he has isolated 2 bacteria which can degrade lignin. And he has obtained another bacteria from a culture collection which cannot degrade lignin. And he has got genome sequenced of all these bacteria, and genomes got annotated, which means genes are marked, and these genes are now available in the gene bank. Each genome has around 5,000 to 6,000 genes, and we want to find novel genes, unique genes, which are present in the bacteria that Rahul has isolated. And those genes will be pinoresinol degrading genes most likely. Next slide, please, Rahul. We have also done a little bit more groundwork, as the experimental crew has told us. It told us to look at the pathway of the compound of interest of lignin pinoresinol. Don't worry about the pathway on the left, but we have elucidated all these steps, how the compound get degraded. What we have found in summary is the pathway was common in the 2 bacteria that Rahul has isolated, and the pathway gave us clues about what type of gene to expect once we have the output from the tools. Next slide, please, Rahul. Because it was the first gene that we were very interested, we did some more experimental work, and we found out that the first step of the catabolic pathway, or the involved gene, incorporates a OH, a hydroxyl group from water, into the molecule. So based on this wet experimental work, we know that the first enzyme we are looking is the hydroxylase. Next slide, please. So now this is the first tool. This is called Unique Gene Finder. What it does is it finds novel genes in an experimental organism as compared to a control organism or organisms. And the tool takes a very simple prompt, as it is written in the green: find me novel genes which are present in X and Y but absent in control. What the tool does, it goes and finds the genomes and the annotations and the proteins, downloads them to the cloud, and runs it. And it gives us a list, which I'm going to show you in the next slide. Rahul, next slide, please. So it gives us a list, an output like this, where the control organism is listed in the red and all those genes are absent. And then it also gives us novel genes which are present in both the other organisms. And now, sorry, previous slide, yeah. Now one of the gene or all of these genes becomes input for the next tool, right? The name and the accession ID of the gene. Next slide, please. Next tool is Gene Whisperer, which takes this protein ID as an input, and it gathers almost all possible information about the gene and the protein from the internet. And then it collates them and gives you a very detailed output. Right now, the tool runs only one protein query at a time, but our next version we are working on, it should be able to run everything that we have written. Like, let's say if there are 50 genes we have found in the first tool, it should be able to run everything in parallel and give you a consolidated summary of what those genes are doing in experimental organism. Next slide, please. Could you show the output, Rahul? Yes, so this is the really detailed report of the Gene Whisperer tool. It tells you everything. It tells about what cofactor the enzymes are using and what domains are present, and if there is anything published in it, everything it collates. If we go back to the next slide again. Yes, and with this tool, the genes that the tool return actually implemented in lignin degradation, and this is a published work, so the tool works. The next part that we haven't published yet, and we want to publish it, what is the structure of some of these proteins and how the substrate fit into it. So we have made a third tool, which is a protein structure prediction tool. It takes input from the Gene Whisperer tool, the last tool. The input is protein sequence in FASTA format, and it creates the structure and shows and gives it back as an output. It is, there it is. Protein, so it takes, and it produces output at a structure. Right now it uses ESM fold program, but we are working on adding AlphaFold, and we are also adding more advanced tools like getting molecular docking done and probably MD simulation at the same time, which will be really useful for taking the project to the next level. I'll stop here, Rahul, and I'll give it back to you.
Thanks a lot for that, Gunjan. And just to sort of take us back to what Stefan described, right? What we want to do with this platform is provide a landing pad, if you will, for all sorts of different tools that scientists use and orchestrate them along with large language model driven agents, so that certain level of automation and agency is actually sort of developed into or built into each and every workflow. And so to do that, there's a couple of other things that we are also working on. Obviously, like Regina mentioned, there's guardrails that we're working on to ensure that the platform actually has a level of security and safety and effective use in line with the R&D policies that we have to follow as a government agency. There is also the concierge or the meta workflows and agents that we are working on, which are going to be released and available in the next release, which are designed to completely sort of act as semi-autonomous co-pilots, if you will, inside the platform, so that they deeply understand what the user is trying to do with the respective project or workflow and actually start recommending things and even co-developing solutions with them. There is obviously a richer interaction with every single output that we have developed. Now, obviously, we've only shown you three examples of 3 tools, but every single tool has a different output. And what we are enabling is essentially every single output should actually be able to explore, the users will be able to explore it with the respective kind of interface. So if the output is an analytical output with lots of figures and tables from a pipeline, let's say for population genetics, then we'll be able to play with it as it needs. The output for the protein structure, for example, in the next iteration will allow users to actually play with the protein structure, interact with it a little bit more, and add more features in the future. So that's sort of, it allows for that level of dynamic sort of UI on the UI end that we are also developing. The next thing that we're developing is data integration, obviously along with the high performance computing and other toolings that are available both within CSIRO as well as outside from cloud providers. And this is critical because we want, now that the base is built, we want to increase the speed with which we bring on new tools, or with which we are able to help develop newer and newer automations or semi-automations with a human in the loop kind of human collaborative approach, and being able to connect all of those data networks as well as those data hubs is actually quite important. The next one that we're actually particularly excited about is also integrating it back into the electronic lab notebooks, because this is built as a sort of sidekick, for the scientists. We want the scientists to be able to use this not only to analyze external information, but also to analyze information from existing records. And not all scientists are sitting in front of a computer. Most of them are in the labs. And so this component is quite critical to us, so that users can also import, not only just import data from electronic lab notebooks seamlessly inside every single project, and that's tied into authentication, but also export these results from this platform into that, so that there is a significantly seamless approach. And what Gunjan did not mention earlier is, this was the example of with lignins that we saw was an actual PhD project. Those 3 steps alone is something that takes a PhD student almost 6 to 8 months to do. If that can't be done in about 20 minutes, then it significantly speeds up the research, and we can actually do a hell of a lot more in that 8 months or the 12 months that a scientist actually has. And to sort of enable that, one of the other things that we'll be working on is shareable workflows, so that if someone actually has a workflow that does really well, that does the job extremely well, then we want to be able to share those across so that others can actually enable them. And this is not just a standard automation workflow. There is a high level of subjectivity in how LLM-driven workflows evolve, because there's a lot of human interaction and human input that we have sort of deeply tethered into this entire interface. And so each workflow will actually be will have a personality. And so being able to share those is actually quite important. Now, we do have two guest speakers as well who will actually, and so I'm going to pass on first to Pete. Pete, please feel free to let me know when you want me to change the slides.
Great, thanks, Rahul. So my name is Pete Thrall. I'm based within the National Research Collections Australia, which is the program responsible for managing CSIRO's natural history collections, primarily focused on characterizing biodiversity within Australia. There's an enormous amount of information that sits within natural history collections, both within Australia and globally, probably somewhere between 2 to 4 billion specimens worldwide, which collectively represent a unique record in both space and time of biodiversity on our planet. Increasingly, these collections are being called on to help tackle a broad range of environmental challenges that are facing us at all sorts of spatial and temporal scales. The problem we have is that most of this data is still actually quite difficult to access, quite difficult to use in research, or other applications, predictive modeling, etc. And this is at the same time as this information is being called on for helping to tackle these problems, those environmental challenges are actually rapidly accelerating. So given the slow pace of species discovery that we currently have, it would take hundreds of years to unpack this information. It's time we just don't have. Next slide, please. So what we need to do is, in fact, exactly what Science Digital is all about. We need to engage with and make use of a whole range of new technologies, including AI, machine learning, robotics, high-resolution imaging, genomics tools as well, and integrate these tools in ways that help us to massively accelerate our ability to not just to do species discovery, but also to build that biodiversity knowledge and to mobilize that data. Digitizing these collections is one of the key tasks that we have to achieve in order to be able to mobilize this data in a way that makes it accessible for researchers and end users worldwide. And it's something that's increasingly being addressed by collections worldwide, but it's still a very labor-intensive, difficult problem, particularly for hyperdiverse groups like insects. There are a lot of insects. They're challenging logistically and technologically to actually image. And so that's what we're really focused on with the Science Digital use case that I'm representing. Ultimately, what we want to be able to do is to then be able to link this kind of biodiversity information with all sorts of other data layers: genetic, taxonomic, environmental, phenotypic, etc. And then to be able to use new analytical tools and approaches to build predictive models and so on to actually start to be able to better understand and predict trajectories like climate change, land use change, biosecurity invasions, and so on, to be able to really develop the kind of scientific impact that collections actually have inherent in the databases or the data that they manage. If I could go to the next slide, please. Thank you. Oops. And so we're really excited about this Science Digital Lighthouse or use case project that we're involved with. The goal in the immediate sense is to really accelerate our ability to digitize and database and make available the data that sits in our insect collections. We have roughly 12 million specimens in the CSIRO collections, most of which is currently not databased, not available digitally. By tackling this problem at scale, we will be able to unlock an enormous wealth of new information. And to do this is going to require many of the sorts of approaches that are being developed through the AI, the Science Digital platform, robotics, automation, deployment of new scanning and imaging technologies, big data processing, and AI-powered tools to help us both capture information from those digitized images, but to also capture the metadata associated with those specimens, which then leads us into an increased ability to do analysis, to do scientific discovery, et cetera. So in one sense, our goal and the thing that we're most excited about is really optimizing and accelerating our ability to deliver data and scientific impact by unpacking this rich biological information that sits within natural history collections. And this is not just a problem for CSIRO, it's a global problem. It's a global problem. And so by solving these kinds of challenges, it doesn't just unlock the data that sits in CSIRO, it actually is going to help us unlock the data that sits in collections globally as well, which is an enormous untapped source of information. We're also equally excited about the fact that this allows us to engage with and be part of the broader goals of the Science Digital platform, the aim to build generalizable tools to accelerate the process of scientific discovery, et cetera. This is something that we're really keen on being part of and contributing to. So we're extremely excited to be part of the Science Digital program. Thank you.
Thank you so much, Pete. I'll pass on to you next, Rob.
Thank you very much, Rahul. Yeah, if you can just move on to my slide. Thank you. So my name is Robert Spate. I'm the director of the Advanced Engineering Biology Future Science platform at the CSIRO. So we're a 5-year, about $25 million project to do fundamental research in the engineering of biology. And you see on the diagram on the right of the screen, we work in 3 main areas. We have a significant socio-economic research component. We do the engineering of proteins, enzymes, strains, and processes, with particularly a sustainability angle. And very relevant to what we're talking about today, we have a big push in what we call data-driven molecular design, where we're really developing a lot of AI and machine learning tools and technologies which are transforming really how we work. We recognized a few years ago how transformative AI and machine learning computational approaches are going to be for biological research and biological design. And so that's why we really brought that big focus onto that. There's really a tsunami of different tools coming at us all the time from different labs and companies from around the world, which doing things which wouldn't have been thought possible a few years ago. Obviously, we heard previously from Rahul and Gunjan around things like AlphaFold, but there's a whole range of tools now that are available that can really revolutionize how we can predict, understand biology and design it and engineering in a different way. So we've got a big focus on that, and we're developing lots and lots of different tools to be able to do that. So we're very aligned with the example that Rahul and Gunjan provide around finding new enzymes, finding new genes, finding out what to do, and then using that to generate processes around that. We also work more broadly in protein and enzyme design, so things like binding proteins for pollutants like PFAS-type molecules, or enzymes that might be able to break down plastics, for example, or be used to make chemicals. So we've developed these tools, these modeling approaches. We also have tools for modeling the cell, so the chemical reactions that happen in the cell. So genome scale metabolic models, which also help us to direct how we might engineer a cell as a production strain for chemicals. But what's really important, as also we heard from Rahul and Gunjan's presentation, is that interplay between the computational side, the dry lab, and the wet lab. So we've had a big focus on developing large-scale protocols that deliver large volumes of high-quality bespoke data sets around the biology. So a particular note, we run a biofoundry here at CSIRO, where we can now generate up to tens of thousands of experimental data points per week on things like enzyme activity. And to really do this well, CSIRO has the ability, and I think it's a superpower of CSIRO, that we can bring together these interdisciplinary teams. And so the work that we do is probably got about 20 to 30 people working between the wet lab and the dry lab, who are working together, say in this interdisciplinary way, to build both the data and the tools and modeling behind it. So despite all of this work, we were really pleased to be able to collaborate with Science Digital, because although we have these tools and we can prototype them, we can test them, although we're generating lots of data, what we really need is a robust platform to host these tools and to be able to interact with those tools and to put these tools in the hands of scientists who are maybe not experts in coding or use of AI. And so for us to be able to have the impact that we want out of our tools, and to have our work have the longevity, and also to deliver this impact into the hands of our industry collaborators, it's really been essential for us to have this collaboration with Science Digital. And we're hoping then, as we build more and more tools into the platform as we go ahead, it's going to be really a powerful way that we're going to interact with other collaborators and with our industry partners, and become really a very general and broad AI for biology type tool, which can push out into lots and lots of different domain areas. And you can see on the diagram on the right, engineering of biology is an essential feature, an essential part of many, many different types of biological research, whether it's in the environment, or whether it's generating new foods, or whether it's addressing health concerns, new forms of energy and fuels that might be generated, or the generation of bio-based chemicals and new manufacturing industries. So I think this is going to be a really powerful tool. We're very pleased to be partnering with Science Digital around this, and we're really looking forward to what this platform is going to deliver. Thank you, Rahul.
Thanks for that, Rob. And I'll hand over to you, Stefan.
Perfect. Thank you. I apologize for this. It's not part of the plan, but here we are. Okay. So now you've seen all this. We asked you at partly ungodly times to join us and dial into this. What are we up to? Why are we so excited to have you here? And how would we like to take this forward? Well, we've essentially asked you to join us and listen in because we want to work with you. We want to hear from you, and we want to do this as part of a community. Most of all, what you've seen here, what we've built, who's using it, how our users are using it, how engineers are building this platform, needs to be educated by real world use case scenarios. At the end of the day, it's scientists who will decide whether using this platform creates the value they want, or whether changes need to be made to the platform, additions need to be made to the platform. This is what we seek to embark on. If you want to foster a trusted tester, contributor, early adopter, and scientific research collaboration ecosystem. Now, you've seen what we've narrowed in with the first instance of the platform MVP in the healthcare and life sciences. And as you, although you've heard several times throughout the presentation that our vision is to bring this to many scientific disciplines, for this first instance of road testing, of expanding and scaling and showcasing the capabilities of the platform, we're looking to team up with researchers and engineers who tackle deep healthcare, life sciences challenges. We also want to work with colleagues who have a substantial track record of publications in top-tier peer-reviewed journals in the field. And very important to say, as it came out a few times throughout this meeting, this is as much a contributor and developer community as it is a user community. So if you are in the business of building AI models, AI agents, if you're creating one or several of those multimodal large biology databases, then that's as interesting as it is if you are a consumer of all of the above. If you're seeking to apply these tools to drive some of your cutting edge research. So that's on a high level the persona of the partner that we're looking to work with. There's a few resources that we have, but that we also would like our partners to have in this. They are around the physical world, around having access to wet labs for validation, compound libraries, biofoundries. And they are around human expertise. So if you have a team that has deep expertise in these fields that you seek to enhance through the use of the platform, the models, the tools, the data in it, then that's a real key ingredient. The scope of the collaborative work that we would like to engage on with you is essentially, well, at a high level, I want to call it decoding biology, decoding life. But we've been a little more specific. So Rahul, Rob, and I have put our heads together end of last year and described our vision for the research areas that we think can be most responsive to being catalyzed and supported and turbocharged with the kind of work that the platform MVP enables. We have summarized that and described that in a paper that you find referenced here and also even linked in here. On top of the technical scientific application-related work, we are also very, very interested in working with our partners on governance of agentic AI networks. You heard us having a strong focus on safety, responsibility. There's also a very strong focus on developing and using benchmarking and performance and impact measuring frameworks, which is a frontier area of research in agentic AI to begin with. And we have, I think, a prime opportunity here to be at the forefront of this. So if you're in that field, we would love to work with you as well. How is this all going to work operationally? So we would like to formulate, explore, and then engage in joint projects. We would like to do work together with you, not just at this point send the platform over and then be detached from it. We'd like to work with you on these aspects of what I've just described before. We would also like to do this in a two-way street. So we really after feedback on using the MVP, what works, what doesn't work. And ultimately, we're doing all this because we would like to showcase how valuable the use of the platform is in scientist hand by creating actual high impact scientific output. And one way, not the only, but one way to measure this is through co-publications. If all this sounds interesting, please do get in touch with us. And we've prepared an expression of interest form that has some of this information that I just ran through and a few more questions that you can reach at this link. And we would really love for you to take a look, to put something in. Please do not share any confidential information at this stage. We appreciate that the circumstances of partnership might be very different from project to project, and we'll work out T's and C's in later stages. At this point, it's really around selecting what is it and who is it that we could work together with on demonstrating, testing, and validating and expanding our platform MVP. So that's what this EUI form will ask you for. It's not too much work, really. A couple pointed questions that will allow us to really pursue this in a prioritized and strategic way, which we will do by pledging to review every such submission that you put in. Nothing will go unnoticed. And then we will get in touch with the submitters of those projects that we think are particularly highly relevant and interesting, and start to explore how partnership could look like. So really invite you all to please have a look at this, think about this. We, just for the records again, we recorded this whole session, and we'll make this available to you, of course, after this event concludes. That will give you access to the links as well in the slides that were shown here. the references, and you have all the information that was shared in this event then for your records to digest, discuss, and include in your considerations on whether this might be something interesting to work with us on. Thank you very much for your attention. I also want to thank all the speakers again. I hope it became clear that there is already a vibrant ecosystem of developers, contributors, scientists, users that works on this platform. We would like to really take this to the next step now that we have a working MVP, now that we can do great work and expand on the great work that you have seen happens in our use cases already. That's the mission. That's what we hope we can embark on. And we're very much looking forward to embarking on that journey with you and your teams. Thank you. That concludes the presentation part. Now, I've seen already during the presentation that many questions have come through. And I can almost tell that we will probably not be able to address all of them in this session. I'll stop talking in a second and we'll revert to the questions then. But I just want to say for the records, all the questions that you have put in the tool, and that, by the way, is the webcast feature, which you find as a little icon on the right side of your screen. The little icon is a question mark. So if you haven't put in any questions and held back, that's how you please can relay those questions to us. So if you don't get to a question that you have put in there in this session, be assured we have monitored who asked the question. We'll get back to you and answer each and single one of these questions. Now, without further to say, I'll take a few of those questions, but then we'll hand over to my team who have all seen those. The first question that I want to address is the following one, and I repeat it here for everyone. What large language models are under the hood, so to speak, behind this platform? Well, there's two places in the platform where those large language models are integrated. It's important to know where they are. So firstly, they are part of the actual toolbox. Right? They might be part of the agents. They might be large language models themselves as tools that agents access. And then there is the overarching orchestration intelligence that decides which agent crews are used for which task and which large language models each of these agents uses. And so the models that we have in there right now are the following ones. So and Royal team, please chime in if I forget someone. There's OpenAI in there. There's Anthropic. There's Gemini, SERPer, NCBI, OpenRouter, Semantic Scholar, Firecrawl, and Brave Search. Do we have any other models that I did not include?
Yeah, just to clarify, so SERPer, Semantic Scholar, Firecrawl, they're sort of services for searching. In a sense, we actually have almost every single large language model technically available because we've set up a proxy server that actually manages the guardrails. But we are triaging all of the large language model providers to only allow and pick providers that actually have the privacy policies that are in line with the requirements for research organizations, such as confidentiality and things like that. So yeah, in a sense, everything, but we're preferring providers that have a privacy policy that is amenable to us.
Very good. Thank you. Which actually, I want to just on that point, that's part where the guardrails come in, for example. That's part where the responsible use of these models comes in and is implemented in our platform, that and other places. Okay. Next question up. Are all the projects in the platforms list visible to everyone in CSIRO? Sometimes there is specific confidentiality, particular for externally funded projects. Right. Currently, projects are private. but all agents are available to all users. So what we are working on currently is to develop a more granular user management system that will then allow for agents that call tools or services to be limited to a subset of users. So breaking this down in more granular access cohorts, which will then, of course, allow to also set the access limitations for agents or models or data that are contributed to the platform, so that they are not necessarily available to everyone else. So there are these levels of privacy and access rights that are implemented in the platform. But right now, there is already that degree of privacy there, because what the project that you start at the very beginning of whatever workflow you then build, that's private to you and you only. Okay. Okay. Next question. I'm interested in exploring how the AI scientific method discussed in the webinar could be applied to strategic direction setting and business intelligence for CSIRO science programs and themes. Is there someone you would recommend I speak with about the feasibility of this approach? So, yes, absolutely. Naturally, and there is a couple of capabilities around brainstorming, hypothesis setting, and the reports that the platform and the findings that it can produce that feed directly into strategic direction setting, making strategic plans, right? Whether you're embarking on a research plan and try to find the gaps or opportunities in the idea that you have, or whether you are embarking on creating a strategic business plan or a strategy plan, and try to identify the market opportunities, try to identify the opportunities for strategic change. That's the same problem set. And so the agentic capabilities that we have in this platform around, I mentioned a few of those agents, brainstorming, hypothesis generation, but also deep research, market analysis, patent search, and others can directly be used to build strategic plans. And so we will get in touch with you separately to point you to the right people on our program with which you can explore this further. Okay, here's an interesting one. How does the platform address AI hallucinations, specifically the issue of generating literature that combines one or more real peer-reviewed articles into fictional references? As I understand it, this has been an ongoing challenge with AI-powered literature searches. Okay, that's an excellent question. And so one disclaimer right up front that I want to point out very clearly. No one has completely and utterly rooted out and solved the problem of hallucinations. Okay, that is just a disclaimer that everyone who is being asked the question needs to be very clear about, and so are we. However, a lot of the research heavy lifting that we have done, that our team is doing, lies in actually building bespoke models, agents, tools that follow guardrails and design choices that minimize and reduce the occurrence of hallucinations. And we're very proud, and we test this all the time, that the degree of hallucinations in our system is absolutely minimal, right? So we can provide you with more information on the actual details of the work that we're doing on building LLMs and models for literature research, LLMs that and models that allow to scrutinize the references that are being used on the way. That's a point you make here in your talk. Like when you have more references and they are peer reviewed, and you are analyzing those to be able to trace what the system has used as it comes to a final output, which of these references have gone in, even which parts, which sections of these references have been used. That's something that we are actively researching. Our innovation team is constantly infusing those findings into our system, and that's what allows us to be saying proudly that our system is not suffering from excessive hallucinations. And having said this, you as a scientist researcher will still have the burden, and that's what our system gives you, the information and material to work with to check what comes out of the system. Right? You can go into the individual sub-steps that the system has gone through, you can check those steps and the outputs, and you can trace this. And you can actually work through this and decide and check that all is as it's supposed to be, and that you're not falling victim to hallucinations. Does anyone else on the team want to weigh in on that one, or just want to open this up to the floor?
Yeah, I just want to add to, along with, kind of the presenting back that factualness and faithfulness, we are looking at other ways that we can incorporate that information into the results people are seeing, such as inline citations, how people can notify or flag for any inaccuracies, but also enabling any user to be able to see the processes that the agents have gone through and where that information is coming from, so that that way they can interrogate those results as well and give it really that critical look through. We do know that this is incredibly important in any results or working with agents, that those results be highly accurate. And so we're continuing to support how we present that back to users and how we verify and validate that information.
Yeah. Could I also add just to that, Regina, that we're also looking at ways that the user can help start evaluating the agent outputs, as you are the experts, and your evaluations on the outputs will be critical. So that's a feature set that we're currently looking at the moment as well.
Yeah, and I think if you look at it a bit more conceptually, essentially large language models are driven by language. They're driven by instructions. And so what we're putting quite a bit of effort into is ensuring that each one of these agentic systems has explicit instructions in every sequential step to critically evaluate the earlier one and then move forward. And that critical evaluation is both based on fact as well as subjective and objective linking between concepts. That's one of the most critical things that we need as scientists. And so we've built essentially a bit of a sandbox that we use to sort of continuously test these so that there is a level of robustness. But I think to both Regina and Pip's point, what we want to do is we want to get as much input as possible or feedback as possible from people actually running these and identifying situations where it either missed something or misrepresented something, so we can actually then go and fix it. And luckily, because of the way the platform set up, a hot fix can probably take just about 24 hours, which is actually quite helpful when we're working on very high value projects.
Cool. Okay, we're nearing the end, but that is a place for a few questions. Let me just scan through which ones I think might be the most relevant to everyone here. Okay, can the platform generate scientific diagrams and or pictures? Right. So the protein structure prediction tool agent is the one that can visualize protein structure. So that's imagery data, and we have plans to develop many more features for visualizations and diagrams. Data visualization is super important, but it does not have that capability beyond that protein structure prediction agent yet. There's a good one. I think that's maybe for Rahul or Gunjan. Can the agent interact with critical third-party bioinformatics platforms such as Geneious or others?
As long as there is an API for it, we can connect it. So the entire platform has been built so that it can continuously route information in and out of any local system or third-party system. And so whether that may currently that is services or providers of certain services or applications like Genius, that is possible. It is also possible if there are any bespoke, for example, large language models in the future that we want to host internally. The guardrails is actually a large language model as well. And so there are a couple of provisions that we have already put in place. There is absolutely no restriction other than the restriction around confidentiality and privacy for services that we can link into.
And maybe the last one for you, Regina, is question 8. I'll read it out, and maybe you can answer. This is fantastic work, and I'm excited to see the integration to coherent and useful user-friendly system. Will you be supporting third-party developers inside CSIRO and beyond in integrating tooling or other AIs via, and then there's a few emerging protocols. So it's a question pointing to the developer ecosystem. I think you would have some thoughts on it, Regina.
Yep, we're definitely exploring how to make that more widely available to everyone. Right now, our
might have frozen.
I might jump in that case, but so I think what Regina was saying, that we are aiming to make that widely available. There's a staged release of certain features. We do actually have
ecosystem. We can understand why we're seeking expressions of.
Sorry, Regina, I think we lost you and then we got you back. Are you able to hear us?
Okay.
Can you hear me?
I think there are some tech problems. Let me just summarize maybe real quick what was the short answer is yes.
Yes.
And the thing to keep in mind is that this is exactly the kind of feedback we would like to seek from working with the community. Like, what are those emerging agent creation networks that we need to connect to? Who's working on them? How can we integrate them? I have a very much like a hugging face for agents kind of vision in my head for what this could, with many more other things. But with regards to the answer to this question, yes, we will support this gradually in a strategic way. I think this is now concluding the time we have on our webcast. So let me thank you, our audience, again for dialing in. Thank you to all the speakers and the team behind all of this. We are super excited to take this further. This is really just the beginning. We are very eager to hear from you what your ideas might be. And as I said at the beginning, we will make sure that no question remains unanswered. Do expect a response from us. Everyone who has asked a question will get that. With that, thank you all. And I'm very much looking forward to further spurring the growth of the AI for Science ecosystem around and with the help of the platform that we built and that we'll test and build with you together. Thank you.